Okay, just a quick question.
Sure.
Yeah, which chapters are we going to cover for this unit?
14 through, I don't remember that. It is on Moodle. You'll see all of the chapters. I want to say there's 5 chapters. I could be wrong. I know there are more chapters than the other units. Let me escape out of this real quick. I'll be able to tell you real quick. I thought I already had Moodle. I already had Moodle on, so just give me 1 second and I can answer that. Okay. Well, I realize now that I open Moodle up that the chapters were out of order on there. That's a slight issue. Okay, I'm stopping that. Let's see. Oops. Actually, I don't have an answer for you right off the top because all of that stuff on there was scrambled up a little bit, and I apologize. I do think there's 5 chapters, maybe. I will figure that out. So 13 really is the one we should be starting with. Not 14, which I had shared just a second ago. Are you seeing 13 now?
Yeah, 13's up.
Okay, perfect. That is where we will start. So, get this. I just try. Okay. So real quick, let me move this around with chapter 13. We are shifting up and we're skipping a handful of chapters. A lot of the genetics stuff I really felt like is kind of beyond intro micro, although some of the biotechnology is interesting. thinking about, how bacteria replicate and how, well, some of those specific details I feel like really kind of is not 100% necessary for shifting it up. We want to think about this next unit is really more immunology. There are a handful of chapters that sort of set the stage, and then we will really get into the details of an immune response. So this unit is basically focusing on that, and then the last one will be infectious disease. There will be all sorts of different organisms. With that said, this chapter does go kind of hand in hand with that as we think about disease, and then in turn our potential to maybe fight that off or the lack thereof. So we will, I will try to make sure that I get these chapters unscrambled on there so that you know which one we're covering first. I thought all that was in order. So I guess before I get going with this lecture, are there any specific questions that you guys have? The exam does close at 5 p.m. today. I see I've got a little more than a third that have completed it. And overall, it looks pretty good so far.
I do have a question about the proctor.
Yes.
So I know that it tracks like movement and whatnot. I'm struggling kind of to find, because I have a Chromebook, which obviously it's not compatible with. If I went to like the Rendezvous computer lab and did it, am I going to get in trouble or docked if like somebody walks behind me or anything like that? Does that matter as much?
It does not. I don't think it's that big of a deal. When stuff like that happens, it like flags stuff, and then the proctor or myself, because I have looked through some of these things, and I get a message from them when it's all said and done. It actually took a little longer than I thought. I reviewed stuff before I ever had the message. But it does look at that stuff, and it flags people if there are issues, if they keep looking away from the screen. There's all sorts of things like that it picks up on, and then I in turn can go back and review that and determine whether or not there were any issues or not. So if you're at Rendezvous and somebody walks behind you, it may mark that, but it's really not that big of a deal. It's not gonna give you a 0 or anything like that. It does not do that. That's my job.
Okay, thank you.
Yep. Okay. Any other questions? All right. So, like I said, shifting it up a little bit, and there's a little bit of jumping around that we'll do in this unit as well, I believe. I don't think it's completely 13 through 18 or whatever it is. I'll have to, again, like I said, I'm going to have to look at that. So with chapter 13, the idea with this, let me get back to this, sorry. It's control of microbial growth is the title of this. And initially, when we're thinking about this, the specifics in this chapter, I oftentimes think about control of growth, and the first thing that really pops into my mind are antibiotics. That is really the focus of the next chapter. This is more thinking other techniques, like when you guys go into the lab, I hope the first thing that you guys do is disinfect your bench. That is kind of what we're thinking about in this chapter. Different mechanisms, like using an autoclave or boiling something, or all sorts of other potentials like that might kill or inhibit growth depending upon the circumstances, not antimicrobials. So we'll talk about some chemical mechanisms, and I believe I should know exactly when all of this stuff is scheduled in the lab, but here in a couple of weeks, you're going to look at some of that stuff where you've got different types of products that might potentially inhibit growth of organisms, or maybe not. And so it's kind of fits with this chapter of material. So in this case, we start off looking at the inside of somebody's car and the potential for, if you were just this kind of reminds me of the very first lab, although you did not have the opportunity to go out and swab whatever you really felt like. You kind of just swabbed a few things to see what sorts of organisms are potentially able to be isolated from the environment. In this case, from somebody's car. And it really kind of makes you start to wonder what sorts of things could you isolate in your own car. I also think about other stuff like, maybe your cell phone. Some of you potentially did swab that to see what was up. So the CFUs are colony forming units. If you think back to growing stuff on a petri dish, those individual colonies, that's what we're talking about. So visible individual or separate colonies is a colony forming unit, the result of a single bacterium replicating countless times to become visible. With that, there are all sorts of areas in this car that they swabbed, and some things a little more, maybe a little more dirty, if you will, or more prone to having bacterial contamination. Things that maybe you touch more. The center console has a boatload of colony forming units, and there's all sorts of possibilities with that. Drinks are placed in there, maybe spilled, maybe providing a good nutrient source for some of those microbes. Then other things, the window control very low, all right? And maybe that's a good indication that your fingertips aren't so dirty. It's hard to say. It really just depends. There's huge variation as far as what was able to be isolated. And this would be the same sort of an idea where you take a sterile swab, moisten it with a little bit of water, swab whatever area in this car, and then in turn, put it onto a petri dish and incubate it up to see what's going on. With that, these colony forming units could be various types of organisms, or it could be a lot of the same. And we're not, at this point, we're not trying to separate and classify any of that stuff. I can't tell you of this 506 how many of those colonies are the same. But as you start, and as you've already looked at a few of these plates in the lab with different types of organisms, hopefully you recognize that colonies oftentimes look different. And that is a mechanism that allows you to kind of differentiate a little bit. So we ended the last unit of material, or almost ended, where we were talking about some of the BSL-4 and working with pathogens like maybe Ebola virus. This is a mechanism that ensures, or hopefully ensures, that the individual is protected from whatever it is they're working with. And in the hood, where they're doing their manipulation of organisms, where they're doing their science, oftentimes we see these as a negative pressure where it keeps the air in, it's kind of sucking in to prevent anything from coming out of this hood. And the suit that they're in is a positive pressure suit, where air is, as you see this red hose coming down, air is being piped into that, inflating that suit. So kind of a double layer of protection with that, the positive pressure in the suit, the negative pressure in the hood, to hopefully ensure safety. There are other possibilities as I'm looking at this hood, where maybe we're not thinking about level 4 pathogens, but maybe you're working with an organism that's an anaerobe, something that can't tolerate oxygen. It might be something that you're also performing under a hood, and oftentimes in these circumstances, they'll have different gas piped in there, that you would be manipulating stuff under these conditions, nitrogen gas or others, but nitrogen is maybe a common one where that hopefully prevents oxygen from getting in. That's not really what we're looking at here, but those are some other potentials. So with that, and we've just saw this BSL-4, which is, as we look at this pyramid, it's the top, the highest risk for serious infection, potential fatal consequences if you're coming in contact with this. So we definitely do not want to expose anyone to this. We don't have at ISU the capabilities or the facilities to deal with any sorts of pathogens like this. We think more level 1, level 2 is what we're working with. Bumping it up to level 3, not so much. I can't, off the top of my head, I can't think of any microbiologist at ISU that's working with level 3 pathogens. We work with some mycobacterium, but not tuberculosis. So bringing it down here to this non-pathogenic level 1. There are a few organisms that we do work with, like Staphylococcus aureus, that are classified as level 2. They do pose a moderate risk. There are some potential issues. With that said, the organisms that we have in the teaching labs are attenuated strains, meaning they have been cultured repeatedly. In the process of doing this, it weakens or dilutes the virulence, the ability to cause disease for these organisms, making them pretty safe to work with, right? That's good. And there's also some potential issues with that because as we rely on some specific reactions for these organisms, sometimes in the process of attenuation appear one way in a biochemical test, and now it doesn't quite work that way again because it's been sort of diluted out, if you will. So there's all sorts of potentials with this, and again, we try to stay within the realm of level 1 or level 2 pathogens, more so with the level 1, trying to keep things as safe as we can. Okay. I'm trying to lecture, and I'm also in the process of being attacked by my house rabbit. He is trying to bite me. So if just bear with me for a second, it never fails. As soon as you try to actually be all business, he's like, nope, I want some pets. Okay, I apologize. Okay, as far as control, and we've dealt with a little of this stuff, like I said, hoping that you come in and disinfect your bench top, hoping you wash your hands, things like that, to help control growth and to hopefully help prevent contamination. We want to know what's on these plates are things that you inoculated and not something that you picked up off of your hand or off of the bench top. So the initial first bit, the first 3 up in here, this is for use on fomites. Oh, as soon as I moved my mouse, I could see the chat. Will we have more chances at quizzes or maybe extra credit to boost our scores? For sure. There will be, and there will be a practice quiz over this unit of material. Definitely. So I'll try to get that stuff posted along with the summary, so you've got ample time to deal with that. Also, Cassie's asking this. She's also said she's got a study group that she is setting up, or a group chat. So if you are interested, look at your chat and you can shoot her an email with your information. Okay, so back to the table. These are a couple of different ideas. So initially, the first three, like I was saying, for use on fomites. Fomites are inanimate objects, things like the bench top or the computer, the keyboard or a mouse, something like that you may come in contact with, but it's not a living organism, right? And so we may treat these things a little differently than if we start thinking about your own skin, right? And oftentimes the level or the concentration of some of these substances is considerably greater than you would think about using on your body, right? So initially, disinfection, this helps to reduce or destroy microbes on whatever inanimate object. And we can think about using heat. Your Bunsen burner could be a potential for something like that. A drying oven, and antimicrobial chemicals. So the disinfectant that you spread onto the bench, I believe, unless they've switched it up, I believe it's called Cytocon. And it works through evaporation. As it's spread onto the surface, it takes a few minutes for it to evaporate to truly do its job. So that's the application, as we said, bench tops, things like that, bathrooms. Chlorine bleach, phenols, Lysol, glutaraldehyde, these are all specific agents that you may use. If you're thinking bleach, it ought to be a 10% solution, not straight out of the jug, right? Dilute it with water, and in turn, that's a very good disinfecting agent. Sanitization reduces the load to safe levels, but we're not thinking about completely eliminating, right? We're inhibiting growth, but we're not necessarily truly killing off these organisms. So that's a little bit different. Commercial dishwashing has sanitization steps to ensure that utensils are safe for multiple people ultimately to be dealing with that. Your own dishwasher at your house does not have a sanitizing step where it's a specific reagent that's applied. So different detergents, although it does say finish here, so maybe finish in your dishwasher does have some sanitizing. I thought that was just to remove spots. So other cleaners and ammonium compounds fit within that. Sterilization, when I think about this, it's more of a kill 'em all sort of approach. It eliminates all vegetative cells. It should also eliminate spores, as well as viruses from that inanimate object. So surgical equipment needs to be sterilized, needles, things like that. Tattoo shops should have a autoclave to sterilize their needles. If they don't have that, you better not be getting a tattoo from that person. Pressurized steam, like an autoclave or a pressure cooker. There's other potentials with some chemicals or radiation, dependent upon the substance. Some things can't tolerate, the high pressure and, well, really high temperature of an autoclave. So radiation might be utilized instead. And there's maybe types of plastics we could think of as an example of something that may not tolerate those high temperatures. Switching it up and thinking about your own body, there's a couple of different potentials: antisepsis and degerming. And it almost seems like they kind of go together. Antisepsis reduces the microbial load on the tissues, and again, some sort of antimicrobial chemical. So cleansing the skin with an injury or before a surgery. Boric acid, isopropyl alcohol, hydrogen peroxide, and iodine or betadine are all potentials to apply to the skin for antisepsis. The degerming is reducing, again, the load, and this is with scrubbing or mild chemicals. Hand washing is an example with that to remove organisms off of your skin. And you did that one of the very first labs when you were looking at some of those potentials. Soaps and alcohol swabs fit with that. I want to bump it up here real quick and just describe describe one little detail about this that I think it's kind of interesting. So sanitization and sterilization, they're not the same. And as I said, with sanitization, you think about inhibiting growth, but you're not necessarily killing those organisms, making it, relatively safe, but the potential might still be there, right? With sterilization, you do think you're eliminating those organisms. So as a microbiologist, I have found myself making some frothy beverages from time to time. Making wine or beer is something that a microbiologist should have some skills to be able to do. So there was an instance when I had some apple juice. We have an apple tree, and we were juicing these apples with, in the back of my mind, what should I do with all of this? Well, obviously, I must turn it into alcohol. That was not what I had heard from the rest of my family. They thought we should have some juice. Why not have juice instead of alcohol? And I thought, okay, I can do that too. So I had all of these wine bottles that I was going to utilize for this. And I made the juice, and I soaked my wine bottles in an iodine solution. As I did this, it sanitizes, it does not sterilize. So ultimately, there were some residual yeasts in some of these wine bottles. And as the apple juice was placed into the wine bottles, I corked them up and put them down in the basement. Some of these bottles, after about a month or so, there was fermentation happening in the bottle because those yeast cells were initially inhibited by the solution, and then as they were bathed in a nice sugary apple juice, the yeast cells started to ferment. So I come downstairs and find a small flood on the floor, but I'm like, there shouldn't be any water leaking here. There's no reason for this. What's going on? And then I turn and see this wine bottle has blown its cork out and has leaked fermented apple juice all over the floor. And then I stopped and thought, why did this happen? It's because I only sanitized, I didn't sterilize. You can take those wine bottles, you can bake them in an oven. So using heat, baking them at 400 degrees for about 4 hours is sufficient to sterilize them. And that was a step I did not do. I had just done this. Hindsight 2020. So interesting. As I looked at this mistake and saw this apple juice that was spewing out, the first thing I did was give this bottle a little smell. It didn't smell too bad. I gave it a taste, and it was a nice hard cider. And there you go. So I evaluated some of these bottles. There were a few that were cloudy and some that were clear. The cloudy bottles were for me, and the clear bottles, well, those were just apple juice. There you go. Let's see, I've got another chat. Yep, see, making beer. It's all good. Yep. And there's all, there are all sorts of things. So after that accident had occurred, that cider was really pretty good. So I did another round where I juiced some apples. I just added yeast without adding extra sugar, and made a very low alcohol cider, which was pretty tasty. My wife told me with that first accident, she's like, wow, this stuff tastes better than the stuff you make on purpose, which really deep down kind of hurt. Okay. So shifting over as we look at this, and we're not looking at a growth curve, we're only seeing the tail end of that curve. What we're looking at here is a death curve. And if we were to see the entire picture of this, when a microbe were inoculated into a broth culture, it would have a little bit of time, a lag phase where it's getting adjusted to its new surroundings. Then it would grow very rapidly, exponential growth. It'd reach a point where it hit stationary phase, where it's, when we think about that in biology, we think of the birth and death rate being equal. At that stationary phase, the growth slows. It starts, it's been accumulating wastes and depleting nutrients. And then you move into this last bit, the death phase, where organisms are dying almost as rapidly as they were growing, all right? So we've got it both logarithmic as a linear bit as we're looking at it, and that's over here on this side of the axis where we've got the number of cells from 10 to the 0 to 10 to the 10th. And then on the other side of the axis, we've got the arithmetic scale, and it looks more kind of like that exponential growth, but in reverse. And we can see this as well as we've got them spelled out. It's the same numbers over here, but just showing what we're really talking about from, billions, 10 billion to, 900 million at the bottom. So the idea with this, the microbial death is logarithmic. It is kind of like exponential growth, really. So we use semi-log plots, and it's easier to evaluate and extrapolate rather than something like this. That's very difficult at this scale with the arithmetic to really know what you're looking at, all right? So whatever it is you're trying to utilize, it's going to vary as far as how this death curve results, but you can see over the course of time in this minutes, so a little less than an hour at 50 minutes, the idea is to bring that load down to, basically to kill them. So time 0, you've got a massive amount of cells, and then after 50 minutes, all right? So the decimal reduction time is the time it takes to kill off 90% of that population. So for whatever that is, and that's what we're looking at right within here, this D value. How long does it take? And you don't need to really get bogged down with that stuff in the first place. Just recognize it is logarithmic, it's exponential when we think about that. So the Bunsen burner is something that you're all familiarized with for sterilizing the loop or flaming the loop, as we say down below. This is one of the key features in aseptic technique. It's something that you do in between streaking for isolation. From the first time you put it onto the plate, you flame the loop to kill off anything on there, back to the plate to spread it out. This super high temperature, turning that little loop cherry red. And as this happens, anything that was potentially on there has been destroyed. Next door to it in figure B is kind of the same idea as this Bunsen burner. This is an incinerator or a bacteriacinerator, whatever, it's an incinerator. And the idea with this, it's a little more contained, if you will. Inside of here, there's a ceramic core, and there are heating elements. This gets red hot on the inside. Your loop would go in here, and any potential pathogen stays within that. So it helps to prevent aerosolization of those possible pathogens. And if you were really working with a, true level 2 or level 3 pathogen, this would be something that you would be more, used more than the Bunsen burner. There are some Bunsen burners that have kind of the same sort of an idea where it's double. There's one bit that's the flame coming up, and there's another part of the Bunsen burner coming off that's sort of like this, but it's using flame in there instead of plugging it in and having electricity heat that up. But it's still, in cases that inoculating loop and keeps any potential organisms from maybe being aerosolized. The way you flame the loop, we hope by going through this practice that it helps to minimize the potential for aerosolization in the first place. But again, serious pathogens, we may think about employing something like this. So this is a schematic of an autoclave. It's similar idea if you think about like a pressure cooker, kind of the same sort of an idea, but this would be something you'd be putting on your stove, or maybe you've got an Instant Pot that similar sort of an idea, but a little more, maybe a little more user friendly, if you will. So here's an actual image of an autoclave, and they are in various sizes. There are some little guys that would sit right on a bench top, and then some big ones like this one that is pretty large scale. I have seen some like I saw one in a hospital that was massive, basically almost an entire room. One room was the dirty room, where soiled materials, contaminated materials go into the dirty room. In that room, there is a door that opens up, and it's a humongous autoclave. You can push a whole cart into this autoclave. You close it, you set the parameters, you cook it up, and on the other side of that autoclave is another room. It's the clean room. And when this is all completed, the door opens up, or you open the door in the other side, and everything in there is sterile. So you're never, in that hospital situation, you're never bringing sterile material out into the same room where there are contaminated materials. It's all kept separate. Oftentimes it's, thinking something like this, there's the potential for that, but usually they're all, everything in here has coverings on it to ensure when it does come out, it's not exposed to the air or anything like that. So an autoclave basically utilizes steam and pressure to get the temperature above 100 degrees Celsius without pressurizing. Once we get to 100 degrees Celsius or 212 degrees Fahrenheit, the temperature doesn't go above that. Spores, like our Bacillus or Clostridium genera, can tolerate those types of temperatures, but they don't tolerate temperatures upwards of 115 degrees Celsius. That's sufficient to destroy those spores. And that's really the whole idea with this type of a technique, to ensure that any sorts of pathogens that could form spores are destroyed. If you are working with something that you're not concerned about that type of stuff, maybe just a simple boiling water, a water bath that's at boiling temperatures, 100 degrees, would be sufficient. So kind of just depends on what sort of substance you're trying to work with. So there's a handful of ways that you can ensure what you have cooked up, if you will, is actually sterilized. Here we've got 2 test tubes, and there's tape on them that is called autoclave tape. This tape, here you see there's no black marks. It hasn't been autoclaved yet. And over on the other side, once it's gone through the autoclave, that temperature causes the specific banding in the tape to change colors. That's a very good indication that what you have cooked up is sterile, right? With that said, there are some potential issues, and we can back it up to this autoclave. Maybe you've got the ability to load this thing up with a handful of flasks, or numerous test tubes. All of these things you're going to be cooking up to in turn use in the microbiology lab or something like that. Well, maybe you have added too many things to this autoclave, and some of it reaches the proper temperature, and maybe some areas in there don't quite get there. The autoclave tape may show that it's been that it's reached that proper temperature, but maybe a few of those tubes didn't actually get as warm as they needed to. You would hope that they all did. So there are some other techniques where you can autoclave, flip back to this, along with your load, you can autoclave a little vial that contains, I think it's a Clostridium organism. It contains some of the spores, and inside of this vial, once it's been autoclaved, if it was effective, it should kill those spores. And if not, once it comes out of the autoclave, you can snap that vial, allowing the liquid in the vial to come in contact with the spore that was also in the vial. They were in separate areas, and it basically inoculates that spore into the broth. Over the course of a day, as it's incubated up, if the bacteria was not killed, if the spores were not killed, you get a color change in the medium indicating that that load was not sterilized as it should have been. Almost always we think about just using this tape, but with some techniques, we may throw that other guy in there just to verify. And sometimes, you know, the autoclave may not be functioning quite properly. A technician may come in and they may run a run through there that uses that little spore, like I said, to truly verify what's going on if it's not getting to the proper temperature as you think it should be. Kind of interesting. So there's some other ideas. Again, depends on the type of substance that you might be working with. Trying to take milk and run it through an autoclave is not the best idea if you want that milk to still be milk. So pasteurization, back to Louis Pasteur, the subject of my coffee cup. Louis Pasteur came up with pasteurization, and this idea, and there's a couple of new techniques with this that we'll see here, but pasteurization basically heats the product, milk or other stuff, up to kind of like up to a steam. We're not bringing it to a boil, all right? Here we say 72 degrees Celsius. Over here, we're going above boiling. And there are others, you know, maybe right at about 60 degrees Celsius or so for just a common type of pasteurization. So you heat it up and you hold it there for a period of time. It depends on the specific techniques, right? But again, typically not thinking above boiling. Some circumstances, yes, all right? But these temperatures that go above, you know, the mesophilic temperature, so like our own types of body temperature or the temperature that lots of pathogens like to reside at, you know, upwards of 37 degrees Celsius, you bump it up above that and it becomes pretty inhibitory. Lots of organisms can't tolerate that. So with pasteurization, Campylobacter was one of the main ones that we really were trying to target in the first place. It causes some nasty GI tract issues. So with pasteurization, you target these guys, but there's others: Coxiella, Listeria, and E. coli, the bad guy, tuberculosis, and Salmonella, and others, all with some rather nasty situations. Pasteurizing the milk ensures that these guys are eliminated. With that said, dependent again on the type of pasteurization, there are some other organisms that may not be targeted by those temperatures. The thermophiles, those that can tolerate higher temperature, can still possibly be there, but we don't think about them as being pathogens, all right? So there may still be some organisms in this circumstance. Over here, shouldn't be, all right? So with that, we've got a couple of pasteurization processes. With this one at 72, this goes for 15 seconds. It's bottled, then refrigerated, and any of these guys eliminated. With the ultra high temperature pasteurization, we bring it up to 138 degrees Celsius, so above typical autoclave types of temperatures. But instead of holding it for 15 to 20 minutes, here it's only 2 seconds. So just a brief, a super high temperature for just seconds, and then it's brought down. It's sealed in airtight containers. And once this is done, this stuff can be safe on the shelf for a couple of months, 3 months or so, without any sort of refrigeration. So you can see some milk that you might be able to purchase right off the shelf, not in a refrigerator. That always kind of makes me feel a little uneasy, but safer, and there are no organisms when we get to these types of temperatures. There's other ideas, maybe using irradiation, where that's kind of almost the same sort of an idea. And similarly, those products do not need to be refrigerated. You can see some of it here in this organic valley type of milk, some of it in the United States where it's just on the shelf. If you go to other countries, if you go to Germany, from what I have been told, I've never been there, but from what I was told, most of the milk, it's not refrigerated in the first place. It's been irradiated and it's just on the shelf. Once you purchase it and open it, you do need to refrigerate it. So kind of crazy. And, you know, thanks to Louis Pasteur, we're able to deal with all of this and not worry about these potential pathogens. So I've got a chat here. Some autoclaves will alarm if they're stacked too full. Definitely. If they get overloaded, if there's too much in there, and yeah, it definitely can sound an alarm to tell you you've got too much stuff in there, but not always. There are some that, you know, you could stuff it full and think you're doing a good job, but you're truly not. You hope your autoclave is actually being effective. So here we've got, and now instead of heat, we've got super cold temperatures. So some ultra low freezers or liquid nitrogen as we look at the other image. This is, although this looks like just a deep freeze that you might have at your house, this is different. This freezer is, as it says, ultra low. Typically, you think about a freezer like in your house getting temperatures to maybe -4, maybe negative 10 degrees Celsius, cold, but nothing like this. All right, the ultra lows going to -70 or -80 degrees Celsius. You know, the Pfizer vaccine, they claimed needed freezing temperatures like this. Now they're coming back and saying maybe that's not true, and I really kind of question why they're saying that in the first place. But these maintain things at super low temperatures, and there are possibilities of specific specimens, medical specimens, or cultures, bacteria, can be maintained at these temperatures. It's not killing them. It basically is, just kind of making them halt their growth. They're in this stasis. They're not really doing anything. If you were going to preserve bacteria, you would mix that bacteria, just like you see the broths and all of these different types of ways to cultivate the bacteria. You would take a small amount of broth culture of that bacteria and add it to a 50% solution of glycerol, and that's going to prevent those cells from exploding once they get to these very cold temperatures. You pull it out of the deep freeze, allow it to come back to room temperature, and now you're able to work with these cells and they're still viable. Liquid nitrogen, this we think of as maybe a more permanent sort of situation with that, right? But there are definitely, tissues that get preserved this way. Flash freezing, it utilizes liquid nitrogen, or maybe not flash, snap freezing, I should say, where it's quickly put into liquid nitrogen. And you've all probably seen some demonstrations with liquid nitrogen where, you put a banana in there and then shatter it with a hammer or something like that. It freezes things almost, well, instantaneously. Not almost, it is instantaneously. let's see, I thought I saw a chat up here. Are we going to cover chapter 14 this week? I hope to get it started. We will see. I don't know. I didn't even look at how many slides were on this, but I do think we will be getting into chapter 14, likely Friday. Okay. Oops. There's that. So this, the Clostridium botulinum, is one of the ideas for why we want to use a pressure cooker or an autoclave in the first place. Botulinum causes botulism. It's also the agent for Botox, for the facelift injected into your skin, and you're now expressionless. These are some serious neurotoxins, and to think about utilizing that in a medical standpoint is pretty impressive. There's other ideas for Botox as well, more than just the expressionless face. It is used with specific things besides that. Excessive sweating could be treated with Botox, and other nerve issues, muscle spasms and things like that may be treated that way. So oftentimes we think about some of these issues with botulism and the improper sterilization. So maybe the pressure cooker isn't working as it truly should. Maybe you're the one doing the cooking. Maybe you don't bring it up to the proper temperature, in turn the proper pressure to achieve that temperature. So as those, whatever we've got going on in these jars, if they're not held at that proper temperature, the spores, which we see right here, don't get targeted. Once the product is brought back down to room temperature, the spore can germinate. So it goes back to this vegetative state. They start growing, replicating, and releasing toxins within that food product. And then you open it up and ingest it, and it's the toxin that's getting you, making you ill, right? So we oftentimes think about things like green beans or something like that that have been, home canning techniques that maybe set the stage for issues. But it's not just that. And there are all sorts of products, if you're thinking food preservation. And with the pandemic, there was a huge boom with that. All sorts of people are like, oh, crap, I better start canning up some peaches or whatever it is. Things like that. Some stuff requires a pressure cooker. Other things that may not have as big of a potential for Clostridium, a simple boiling water bath is sufficient to ensure that those things are preserved. Oftentimes having stuff like, more sugars, if you're thinking jams and jellies, things like that, don't need a pressure cooker, just a water bath is sufficient to inhibit any organisms. There's other ideas with that as well that we might think of for preservation. And again, the preserves, as I mentioned. So adding sugars to this creates a situation where it becomes very hypertonic. So if you've got a cell, a microbe within there, it pulls the water out of these cells, it dehydrates them, and it in turn kills them off, making it safe, right? Relatively safe, I should say. So some dried directly. We've got a little bunch of raisins right here, jerky, as another potential through drying. There's also possibilities of using salt. Salting out something helps to do the same thing, pulling all of the moisture out. Salted fish, the addition of sugars with jams, all of these set the stage to preserve. There are some potential issues with this, and some of these things may inhibit bacterial growth, but sometimes something like maybe like a home, a freezer jam or something like that, maybe it's not, if you're making it at home, you're not adding other preservatives and things like that into it. And sometimes fungi can get in there and tolerate, even though the concentration is really strong for the bacterium. The fungi are like, oh, this is fine, and I can still grow. So sometimes you might see the jam getting some mold in there. Jerky might do the same thing. If it's not refrigerated, it could potentially start to get moldy. And there, definitely some issues with that. And then for the most part, we're thinking about, inhibiting or preventing these microbes from getting in and really causing big issues. There are some circumstances, the salted fish, maybe dried fish. There are some parasites that maybe in this process, it doesn't actually impact them, and consumption of them, you can still ultimately get ill from that, but that's relatively rare. So we won't worry about that for right now. Another potential for mechanism is the UV. Oh, I just realized we're like 5 minutes past. Somebody should have piped up and said, "Hey, shut up." That was really probably like, are we getting to 14? I apologize. I'm going to stop right now. There it is right there. I think we're going over time. It's because I wasn't paying attention. I was just getting all excited. I apologize, guys. I will quit right now. We'll pick up with the rest of this Wednesday. Okay.
Thank you.
Questions or concerns? Everyone's like, I've got class. Here, I'm just sitting at my house. I'm like, I guess I'll fine and good. I'll do this whole chapter right now.
Thank you.
Thank you. Have a great day.